Okay, hey people. It's time to start metabolism. We're gonna be in metabolism for the whole rest of the semester. So this is an introduction to metabolism video. It doesn't have any specific pathway in it. It just has some general ideas and themes in metabolism. So after watching this video, you're able to define key vocab terms, draw the mechanism of ATP hydrolysis with a corresponding reaction coordinate diagram. Describe why ATP is the energy currency of the cell. Everybody always talks about ATP being the energy currency, but I wonder if you know why or if you're able to describe why. Describe 3 sources of ATP. And then we'll talk about NADH. So you should be able to describe the function of NADH, I'll say with respect to metabolism. Okay. Recognize the structures of NAD plus and NADH and determine whether a molecule is reduced or oxidized in a chemical equation. Okay, so all living cells need energy to grow and to move. And this energy to build the cells and to move comes from chemical reactions that happen inside of the cell. And some of them happen outside of the cell, but we'll mostly focus on ones that happen inside of the cell. And there's 2 really broad categories of chemicals that happen, reactions that happen within a cell. And those are ones that are catabolic, that describe catabolism, which is where you take a complex molecule and you break it down into something that's very simple. So an example of a complex molecule might be a tag, and that could be broken down into the simplest form of carbon, which is CO2, the simplest form of an oxidized carbon, I should say. Another complex molecule that you've met is glycogen. And again, to break that down into the simplest carbon is again CO2. And so I picked these 2 because tags are your fat storage and glycogen is your sugar storage, but in total, they comprise the reduced carbon storage of your body. Okay, so what do we mean by reduced carbon? So a reduced carbon has a CH bond, and an oxidized carbon is sort of what it sounds like. It's a carbon with an oxygen. So that's what we describe as reduced and oxidized in biochemistry. Please note that this is going to be different if you're in Gen Chem or inorganic chemistry. And then anabolism is just the exact opposite of that, is where you take a simple molecule and build it up into something that is complex. And so I'm actually not going to go backwards from CO2 to glycogen because only certain organisms will do that. So if you went from CO2 up to glycogen, only the autotrophs will do this. And an example of an autotroph is a plant. Okay, humans can't go from CO2 to glycogen, otherwise we wouldn't need to eat, we would just breathe in the air and become giant pumpkins because glycogen takes up a lot of space. But instead we take molecules like pyruvate, which I'll just tell you is 3 carbons, so it's rather small, and then we can use that to go to glycogen, which if you remember can hold up to 50,000 glucose units. So again, a really teeny tiny guy to a really, really big one. And you can also go from pyruvate to a tag as well. Okay, so catabolism and anabolism. In this diagram right here, catabolism is in red, going on down, and then anabolism is coming back up here. So they're examples that they have in this diagram of complex molecules are proteins, nucleic acids, polysaccharides, and complex lipids. I only grabbed the polysaccharides and the complex lipids. You don't break down nucleic acids for energy, and proteins store a lot less energy and have some other functions. Okay, so the breakdown of a tag isn't just going to occur. Breakdown of tag to CO2 or glycogen to CO2 isn't going to happen in just one step, in one reaction. There are many, 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 many steps. And all of those steps are housed within central pathways. And central pathways account for large amounts, or the largest amounts of mass transfer and energy production. These are where there's the heaviest traffic, and they are also very highly conserved across organisms. Okay, so there's a lot of pathways that are in this particular diagram here, but there's a couple that I'll highlight. So you have glycolysis here, and again, the red arrows going down are your catabolic, and the blue arrows going up are your anabolic. So the opposite of glycolysis is gluconeogenesis. Those pathways are running in opposite directions. We also have the citric acid cycle. You may know this as the Krebs cycle or the TCI cycle or the tricarboxylic acid cycle. So many different names for the exact same thing. We have a process down here called oxidative phosphorylation. There's photosynthesis. And then, of course, there are a number of pathways over here, over here, over here, that all basically feed into these central pathways. And I think the most central pathway is this one right here, the citric acid cycle. And this is because this is where the CO2 will come out. And if you've noticed, I have a lipid going to CO2, I have glycogen going to CO2. So we know that we'll be done metabolizing something or breaking down a molecule when all of its carbons, all of its CH bonds have turned into CO bonds. And the most oxidized flavor of carbon in your body is CO2, and that is the major carbon output of the citric acid cycle. So all the other pathways have a lot of intermediates that are not CO2, but the citric acid cycle, its product, its carbon product is CO2. So you'll notice that all pathways basically either lead to acetyl-CoA, or they lead into glycolysis or gluconeogenesis and then feed down into that pathway. So anyway, so central pathways account for the largest amount of mass transfer and energy generation, heaviest amount of traffic. Again, all roads lead to acetyl-CoA or to the citric acid cycle, and they're highly conserved across organisms, which means that you can find the citric acid cycle in just about every single organism on this planet. Not all of them, but almost all of them. Okay, so there are separate pathways for biosynthesis and degradation. So that means that there's a separate pathway that will break down a fatty acid versus the pathway that will make the fatty acid. So here, this is catabolism, and here, this is anabolism. And there's a few different reasons to have separate pathways for anabolism and for catabolism. One is that a pathway must be exergonic, negative delta G, overall, in the direction that it proceeds. Okay, so that means that going in this direction, we have to have overall a negative delta G, and going in this direction, we have to have, again, an overall negative delta G. So you can't have it just be one single pathway that goes the exact same enzymes for both of them, because they couldn't both be negative delta G. And in order to get your pathway to move in a forward direction, it has to be exergonic. So we have separate pathways for catabolism and anabolism, even if they have the same intermediates. This is an intermediate. We may also call this a carbon metabolite. Okay, and we'll talk a little bit more about some examples of intermediates and carbon metabolites. This is also an intermediate or carbon metabolite, because these carbon-based molecules go on to do other things than just be part of these pathways. Okay, and then another reason that we have to have separate pathways for catabolism and anabolism, even if they involve the same intermediates, is that they have to be regulated. Okay. Regulated, let's say to meet physiological needs. And this essentially, it keeps the pathways from being futile or having futile cycling. Okay, so this diagram that I have drawn all over and looks like a giant hot mess at this point is a futile cycle, meaning that in one direction ATP goes in and the other direction ATP comes out, and overall there's no net generation of ATP in either direction. And so it goes around and around and around and around again, and being able to regulate the pathways to meet physiological needs that basically you don't want to break down a fatty acid at the same time that you're trying to make a fatty acid. That's essentially the rules of regulation and futile cycling. Don't want to break down a fatty acid at the same time you are making a fatty acid. And we'll come back to regulation once we get to the end of metabolism. Okay, so one goal of metabolism is to make ATP. ATP stands for adenosine triphosphate. And I have a molecule of ATP here. You don't have to be able to draw this. You should recognize it though. And it has 3 phosphates. That's why there's a tri. And the bond that connects the phosphates is called the phosphoanhydride. The phosphate that's closest to the sugar is the alpha, and then beta, and then gamma. Just food for thought. So why make ATP? What is it good for? So ATP is particularly useful, yikes, energy is created or generated when ATP is hydrolyzed. Oh dear, there's that word hydrolyzed again. Okay, hopefully at this point you are seeing a pattern. Hydrolyzed means we're going to break a bond, or we're going to break apart the molecule. We'll actually create a bond, but we're going to break apart the molecule using water. So and the bonds that we're going to break are the phosphoanhydride bonds. Okay, so the hydrolysis of ATP releases energy. We're going to draw this mechanism out, and we're going to do a reaction coordinate diagram, and I'm hoping that you'll be able to explain why it is that ATP releases energy when you break it, and then obviously it's the exact opposite when you have to make it again. So I'm going to just draw the phosphates out. Triphosphate, and then I'm going to put an S for sugar and a B for base. So this is the sugar base. So this is a nucleotide because it has a phosphate, a sugar, and a base. For whatever reason, I always put the double bonds up, and then you just finish surrounding them with oxygens. Okay. And again, with hydrolysis, I'm not going to draw the enzyme catalyzed reaction because it just gets a little bit messy, but you can imagine that this happens inside of an enzyme. With all the residues surrounding it in the active site. Okay, so I have my oxygen, or I have my water that has a nucleophilic oxygen. Again, there is a partial negative on that oxygen, and since it's biochemistry and it's in water, we can always have another water come and grab that hydrogen to make it an even better nucleophile. So now that instead of a partial negative, I would have a full negative. And here on the phosphates, all of the phosphates have a partial positive. So again, opposite charges attract. So this nucleophile will attack that electrophile. Electrons go up, they come back down, and we're going to kick off the diphosphate, or the dinucleotide. And the products of this are what's called a pyrophosphate, or an inorganic I'm sorry, not a pyrophosphate, an inorganic phosphate. A pyrophosphate is 2 of them. Okay, this is an inorganic phosphate, sometimes abbreviated as PI for inorganic phosphate. We have an extra hydrogen, and then we have our ADP, because there's 2 phosphates, D for diphosphate. Okay, so why is it that the hydrolysis of ATP releases energy? And please don't tell me it's because you broke a bond. And the reason you can't tell me that is because, yes, we did break a bond. Which color should I use? Yes, we did break this bond, but hey, check it out. We also made a bond. So this whole idea that making or breaking bonds, like is a general rule for whether energy is released or not, is probably not the best explanation. Instead, I want you to think about it. Remember our handy-dandy reaction coordinate diagrams? This is reaction progress. This is not time. This is not time on the x-axis. It's just reaction progress. And then I'll have energy over here on the y. Okay, I know that going from my reactants to my products is a negative delta G. Okay, which means that on a reaction coordinate diagram, my products have to be lower in energy than my reactants. So if I'm going to give this values, we'll just say the units over here, kilojoules per mole, and I'm going to say this is 50, and I'm going to say this is 100. Then going from here to here, final minus initial would be a negative 50 kilojoules per mole, and that makes this whole thing exergonic. Okay, and again, I could draw in a transition state. I don't know how high the bump will be. But why is it that my products are lower in energy than my reactants? So that's why this is exergonic, is because my products are lower in energy than my reactants. Hopefully you can see that. 50 is a smaller number than 100, and I just picked those random numbers. So this is ATP up here. This is ADP, PI, and that proton. So why is it that my products are lower in energy than my reactants? This is why I have a negative delta G. So there's a few reasons. Okay, the first one is that in the products, so these are the products right here, there's a better separation of the negative charge. Okay, so if you notice in ATP, we have a lot of negative charge in a really teeny tiny space. And in ADP, we've separated those charges out, so the products are, in this case, that makes them more stable or lower in energy. Hence, this bar is lower than that bar. Okay, there's another reason that our products are more stable, and that's because we have more resonance when we separate them out. So the products have more resonance, which is stabilizing. They have better charge separation, negative charge separation, which is more stabilizing. And if you'll notice, just overall, we have 1, 2, 3 products, and we had 1, 2 reactants, so we have more products than reactants, which is a favorable entropy. Okay, so more stable because we have separation of the negative charges. Our products have more resonance than our reactants, and we have more products than we do reactants, so that's a favorable energy. We've created more stuff, more chaos. So all of that contributes to this bar being lower than this bar, and that is why when you hydrolyze an ATP, sorry, I should have put water over here. Forgot that. That's why when you hydrolyze an ATP, it releases energy. The products are more stable than the reactants. They're more stable because of charge separation, more resonance, and you have more products than you do reactants. So I'm hoping that you'll be able to draw this mechanism out and be able to explain in terms of thermodynamics why hydrolysis of ATP releases energy, and it has nothing to do with the fact that we broke a bond, because we also made a bond. Okay, I found this slide, and I thought that I would just throw it in here because I know that it's been a while since you've had organic chemistry, and we've been through a few mechanisms. So these are some common electrophiles, and I tried to highlight this in the previous mechanism, but if you'll notice, your electrophiles will always have a partial positive on them somewhere. And in biochemistry, it's pretty much always the carbonyl carbon. So carbonyl carbon is an excellent electrophile, has a partial positive. A protonated imine, we don't see that too much. And then again, the phosphate has a partial positive on the phosphorus there. So those are all great electrophiles, meaning they can be attacked by a nucleophile. So for electrophiles, you're always looking for a partial positive atom. And then the nucleophiles, you're looking for something that has a partial negative or a full negative. Typically, we're using hydroxide ions, which may come from water. I'm trying to think of some other ones that we may have used. So far, we've only used the hydroxide ions, but so you'll notice that the nucleophiles, they either have a partial negative or a full negative charge. That's just a reminder. Okay, so ATP can be used to drive chemical reactions that would otherwise be thermodynamically unfavorable. So let's say we have a reactant that goes to products, and it has a delta G of, I don't know why I had to put the plus there, of let's say positive 40 kilojoules per mole. So this is an endergonic or unfavorable reaction. You can couple this with ATP hydrolysis. Sorry, this is a PI, where the delta G here is negative 50 kilojoules per mole. And if you couple those reactions, which means they happen together, or you add them together, you combine all of your reactants. And then you would add the delta G's together. So overall, the delta G of this, when I add them together, is a negative 10 kilojoules per mole. And that coupling makes this whole process exergonic and spontaneous. It doesn't tell you how fast it goes, but it is going to release energy when you couple them together. So that is the idea with ATP hydrolysis coupling, is that you can drive unfavorable reactions forward or towards the product. Okay, so oftentimes we call ATP the free energy currency of this, and the currency really does just mean that ATP energy can be, or let's say ATP hydrolysis energy, so the negative 50 kilojoules per mole, that energy can be exchanged or coupled to an endergonic reaction, just like the one we just did. Okay, so why ATP? Why not GTP, UTP, DCTP, or any of the other NTPs, where N here is any base? Why is it ATP and not any of the others? And there's really no particular reason. They all release about the same, they all have the same delta G when they, I can't speak. They all release about the same amount of energy when their phosphates are hydrolyzed. But it turns out that evolutionarily, wow, yikes. I don't know why I bother trying to spell. There's just a higher concentration of ATP than the other NTP. So in the cell, ATP is about 3 to 5 millimolar, where the other NTPs are about 1 millimolar or less. So there's just more of ATP in the cell, and so it's used more frequently. And then the enzymes have evolved to recognize the A, the adenosine, and not G or C or U. So there's no very particular reason other than evolutionarily enzymes have evolved to recognize this ATP, and therefore there's also a higher concentration of ATP than the other NTPs. Okay, so what do you use it for? So I said in 2 slides, or 2, whatever, I don't know if you see these as slides or if you just see this as like a rolling video. But in 2 previous statements, I've said that you can couple it to unfavorable reactions. And so one that we've seen is that you can couple it, well, you cannot see that green. You can couple it in active transport. So the ATP that's made during metabolism, you can couple it to moving things across the membrane. You can also couple it to myosin or your muscle contraction. So you need ATP. Can you see that there? Why do I keep picking the wrong colors? You can couple it so that the muscles can move. So ATP is used for a number of actual examples, not just reactant goes to product, which is unfavorable. But in this case, we have an unfavorable movement of ions, and that can be coupled to ATP hydrolysis. The movement of myosin is unfavorable, and you can couple that to ATP hydrolysis to get it to move forward. And so now we're going to talk about a little bit about how do living things make ATP. There's basically 3 ways to make ATP, and we're going to focus heavily on 2 of them. So we're going to focus a lot on the first 2 and not at all on the third one. So substrate level phosphorylation is where a higher energy compound is broken down. So you get the breakdown of a high energy molecule to a lower energy one. So something like a phosphoenol pyruvate to a pyruvate. I know that you don't know what these are yet, but we'll talk about that. This is higher in energy than pyruvate is, and so we can get an ATP out of it. So in this case, PEP to pyruvate is a negative delta G, and that can be coupled to the positive delta G of having to make an ATP. So this is one way to make an ATP, is something that's higher in energy is coupled to something, higher in energy is broken down to lower in energy, and that can be coupled to making an ATP. So if making an ATP in this case is positive delta G, again, if you break it, or if you hydrolyze ATP and that gives you a negative delta G, then when you synthesize it, it has to be a positive delta G. Those are opposite reactions. So oxidative phosphorylation is, I'll just call this favorable redox coupled to ATP synthesis. So in this case, the redox has the negative delta G, and again, the ATP synthesis is the positive delta G. And you can couple those together. So this one is literally bouncing electrons around, and those electrons go in a very favorable direction. It releases a lot of energy, and that can be coupled to, can you even tell that this is plus delta G of ATP synthesis? And then the last one, photophosphorylation, is just you use the energy of light to synthesize ATP. Okay, so like I said, we're going to talk about substrate level phosphorylation quite a bit. Substrate level phosphorylation is our catabolic pathways. And we're going to be wow. Hey, man. Hey, shrink. There we go. So in the catabolic pathways, we're going to be talking about breaking down organic carbon or reduced carbon, which always has a CH bond, into oxidized carbon, which is a CO bond. Okay, and the breakdown of organic carbon into inorganic carbon is heterotrophy. So heterotrophs, they go from organic carbon to inorganic carbon. And again, by inorganic carbon, I mean a carbon that has an oxygen attached to it, and our organic carbon has to have a carbon-hydrogen bond. And heterotrophs go in the opposite direction. So I've got this diagram here. It connects heterotrophy with autotrophy. So here is a polysaccharide. Sugars have carbon-hydrogen bonds. That makes them organic. Breakdown, 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 breakdown. Why am I using the same color that's in this? Breakdown, 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 all the way to the inorganic carbon. And that inorganic carbon is used by autotrophs to make organic carbon. And that organic carbon you eat, and it is made into inorganic carbon that the autotrophs then use to make organic carbon, and then you eat the inorganic carbon. Basically, autotrophs, they make their own food. And the heterotrophs eat the autotrophs. Or they eat the food that the autotrophs made. Okay, so an example of an autotroph is a plant. Some bacteria are also autotrophs. But we, as humans, we're heterotrophs, and we're going to focus on heterotroph metabolism. If you want to learn about autotroph metabolism, take microbiology. It's a pretty cool class. Some examples of organic energy sources. Again, these things have to have carbon-hydrogen bonds in them. So you have your fats, your triglycerides have lots and lots of carbon-hydrogen bonds, so they store lots and lots of energy. These are all sugars here. Again, they all have carbon-hydrogen bonds. They have less of them because they have some oxidized carbons in them, so they store less energy, but they still have lots of carbon-hydrogens. Proteins have carbon-hydrogen bonds. And then there are some organisms that can eat these other weird molecules that we can't, that we might consider toxic. But like bacteria, they can break down some of these polychlorinated aromatics because there are carbon, actually there's not carbon-hydrogen bonds in this one, but there's carbon bonds in them that can then be reduced and then oxidized. But there's carbon-hydrogen bonds here, carbon-hydrogen bonds here that can be used for energy. So what's so special about the carbon-hydrogen bond? Why do we care so much about this? Well, the carbon-hydrogen bond is the one that's holding all of the energy. So if you think about the most reduced carbon, methane, and if you light methane on fire, you get this boom, this really big reaction, and it releases a lot of heat energy. But what comes out of it is carbon dioxide, and I know there's a few other things going on here. You need oxygen, and that goes to water. But we're looking at the carbon, so we're looking at the connection here from carbon to carbon. So all of the carbons, the carbon-hydrogen bonds, go to carbon-oxygen bonds, or the most oxidized carbon. So what I'm trying to tell you is that there's a lot of energy stored in a CH bond. And there's not a lot stored in the CO bond. So all of our foods that we eat, when we eat them, are going to go to CO2. You will know that we're done with all the pathways completely metabolizing a carbon if we make CO2. Okay? And in the process, when we release this, so you don't have little fires going on inside of your cell. That's just not happening. You're not on fire on the inside of your cell. Instead, on the inside of your cell, you have something like glucose goes in, many steps, and then eventually CO2 will come out. But you know that when you do that, when I go in that direction, I should be releasing a lot of energy, and that I'm oxidizing my glucose. if I'm not on fire, then that energy must be transferred somewhere. And the energy is transferred to these electron carriers, like NADH. So instead of being on fire while glucose is being oxidized, the electrons are transferred to what are called electron carriers. And they may also be called reduced electron carriers. Okay, and so these are carrying electrons, and we'll meet one of them, and it's going to deliver these electrons to an electron transport chain. And those electrons in the electron transport chain are going to be coupled to making ATP, and that is the oxidative phosphorylation. We also are going to get some ATP, some substrate level ATP coming out of here. So let's meet NADH. So NADH is called a coenzyme. Coenzymes are organic, which means they have carbon-hydrogen bonds, versus a cofactor is inorganic. So an example of a cofactor would be magnesium. It's not organic, but it might help the enzyme function. So NAD stands for nicotinamide dinucleotide. I'm hoping that you can see that there is 1 and 2. Those are the 2 nucleotides. This pink region up here is the nicotinamide. So this is a 2 electron, 1 hydrogen carrier. Go back. Oh crap, I don't know if I went back or forward, so I don't know what just happened there. The 2 electron, 1 hydrogen carrier. Here, where's my NADH? Okay, so my NADH is more reduced, is because it has an extra hydrogen, or it's carrying the electrons, has more electrons. Hopefully you remember your oil rig, oxidation is loss, and reduction is gain. So NADH is the reduced form. NADH is the electron carrying form. And then NAD plus, because there's a little plus sign right here, it's an ion, is the one that's going to sponge up the electrons. So NAD plus is the oxidized version, and NADH is the reduced version. And it turns out that we just met a vitamin. So NAD is formed when you add vitamin B3 to it, which is also known as niacin or nicotinic acid. I'm sure I spelled that right, or spelled that wrong. So this is vitamin B3, and you'll notice that it's this unique base up here. It's not A, it's not G, it's not C, it's not U, it's not T. It's nicotinic acid or niacin, and this is the functional group that's able to accept the electrons and lose the electrons. So just so you know, keep track of your vitamins. We're going to meet a whole bunch of vitamins. And also just another food for thought is that NADH carries about 3 times the energy of an ATP. So it's very wise, or it makes sense that the cell would be carrying a lot of energy around an NADH because you don't need as many of them as you would need an ATP. So you need 3 times as less of an NADH than you do ATP. And then you have this energy source just kind of wandering around the cell that can be used to make ATP. Okay, so why do some enzymes require coenzymes, cofactors, or prosthetic groups? Remember, an example of a prosthetic is a heme. An example of a cofactor might be a magnesium. An example of a coenzyme is NADH. What makes a prosthetic group a prosthetic group is that they tend to be covalently attached, whereas the cofactors and the coenzymes are very weakly bound. So this is the non-covalent forces that are holding these together. And I do have to say something. A lot of biochemists use the word cofactor to mean NADH and magnesium. It's kind of a catch-all phrase, and very few will actually use the word coenzyme, but they are different things. And for the purposes of this class, we will call NADH a coenzyme because it's organic molecule, and we will call things like magnesium or iron or copper, things that are not organic, things that don't have CH bonds, that are required by an enzyme to function, we'll call them cofactors. Okay, sorry about that, too long-winded. So the coenzymes and the cofactors tend to have something to do with redox, and the reason that they need them is that the amino acid side chains are just not good at accepting or donating electrons. They're just not good at it. The same thing with hemoglobin. You have to have the heme because the amino acid side chains are not good accepting that oxygen. Okay, so I have to do an example because I want you to see some of the concepts. So this is a chemical reaction for the glycolysis pathway. Okay, where we have glucose going to pyruvate. These are our carbon intermediates. You may also call them the metabolites. Okay, so glucose has a lot of CH bonds. There's one here, 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 here. There's two here, and another one there. And then pyruvate, it has just 3 of them here. So we're going from something where glucose is more reduced compared to pyruvate here is the more oxidized of the pair. So when you compare glucose and pyruvate, you can see that we're on the way to getting to CO2, but we're not completely there. So glycolysis is not the end of the line for breaking down a glucose, because we have to get from pyruvate to CO2. So hopefully you can see that pyruvate is not CO2. 1, it's not called CO2, and 2, it's not carbon dioxide. But we have broken down some of the carbon-hydrogen bonds, and they are being moved from an NAD plus to an NADH. And again, the NADH here is more reduced than the NAD plus. So the electrons are going from glucose to NAD plus, which creates an NADH. And in the process of glycolysis, we actually also get some substrate level phosphorylation. This is not oxidative phosphorylation, but this is substrate level phosphorylation. And there are 10 steps. There are 10 steps in glycolysis. I'm just going to do a mock pathway with 1, 2, 3, 4. I have 5 carbon intermediates. Okay, so here's a mock pathway with 5 carbon intermediates. The first ones and the last one do count as intermediates because they're likely going on or coming from other pathways, going on to or coming from other pathways. Within the pathway, we'll have some, one of the steps will be rate limiting. I'll just say that this is the rate limiting step, or the slowest step. If this one is rate limiting, then that means that this intermediate accumulates. If there's an equilibrium here, then this one will also accumulate. So anything that comes before a rate limiting step will accumulate. It's just another chemical logic of the pathway, and there'll be a reason to accumulate these intermediates before a rate limiting step. This step also happens to be the, what do I want to call this? The point of regulation. I'll call it the major point. Okay, that was a whole lot. I have no idea how long this video is. I'm really sorry about that. But enjoy your first day of metabolism, and I'll see you later.